Hi, welcome to this video. This is a lecture originally prepared for the iGEM Spring Course in 2019, being recorded here for the benefit of the 2020 team. In this video, I'd like to go over how you can take information on BioBricks and start to assemble different types of circuits. And I'll go over some common motifs and logic gates that are found in circuits in synthetic biology. So these different genetic circuits are built using different DNA parts or BioBricks. These are standardized DNA parts which you can find cataloged at the iGEM registry. These different BioBricks can be assembled together in a systematic way. So you can find different BioBricks or composite parts and assemble them together into even larger parts. And depending on your method of assembly, you could continue this, in theory, to an infinite size, although there are some practical and concerns and physical constraints you'll run up to. But the idea here is that we can think about these parts in an abstract way. We don't have to worry exactly about how the sequences are going to be fused together. These different BioBricks, and there's a lot of them, over 20,000, can be found again at this registry. The website for this is parts.igem.org. We have many of these parts, or at least some of the best characterized ones, available in the lab today. So when we talk about a genetic circuit, what do we mean? And why are we going to worry about engineering these? Well, by taking together different parts and stringing them together in a particular way, we can program sophisticated behaviors into a cell. In fact, a lot of the terminology and the thinking behind how this is done originates from fields of electrical engineering. So you'll actually see some terminology and diagrams here that are borrowed, like this one, borrowed from a legend for, let's say, an electrical circuit diagram, where we won't have things like diodes and capacitors, but we will have logic gates. To start to think about how we're going to apply this abstract thinking and engineering paradigm to genetic circuits, consider what we have at the bottom of this slide. We have input molecule A, which activates gene expression C. A very, very simple genetic circuit. The more of the input A we have, the more of the output C we get. This could be a very straightforward type of regulation. We can have an activator protein, and the more of that activator protein we have, it binds to the promoter of gene C, and we now get more of the output of C, which let's say is green fluorescent protein. So a very straightforward example. Example of this would be what you can find in quorum sensing, where you have a promoter which responds to a combination of a transcription factor and a small molecule, a quorum sensing signaling molecule. And when these two are present together, they will activate gene expression. Let's say we want to take that behavior and flip it on its head. We would need to include an inverter. So an inverter would change the gene's behavior. So instead of A leading to an increase of C, we would see that addition of activator A would actually decrease expression of the target gene C. How would we do this? How can you accomplish this effect? Well, an inverter usually is the addition of a repressor gene in the middle of this genetic circuit. So instead of activator A binding to gene C's promoter directly and activating transcription, it instead will bind to the promoter of gene B, activate and create gene B, and B here is a repressor which will shut down C. So by combining different genetic modules, we can change the behavior here of this circuit. And when creating these circuits, it's not always necessary to get into the nitty-gritty of understanding how all of this works. If you have a part that's labeled as an inverter, you simply insert it between the two other parts you have, and you more or less will get, depending on how those parts are set up, you should get this kind of behavior. Here is a A specific example, if you take something like the Lac Operon, normally Lac I will inhibit Lac ZYA, and then you would have something like IPTG, which could relieve inhibition. If we instead change the circuit so that we have a repressor protein like C1 in the middle, we will have inverted it. So now, when you add the inducer, IPTG, you will be able to create more of CI. So you can see here in this graph, CI production goes up. But since CI is a repressor of Lac ZYA, the level of that protein will actually go down, which is the opposite from how the Lac Operon normally behaves. Let's look at some other simple logic gates. So we already saw how introduction of another set of transcription factors, a repressor and its associated promoter, can create an inverter. Here, let's look at an AND gate. So for an AND gate, you will only get the output C when you both have input A and input B. Low amounts of either of those will lead to no output. An AND gate can be useful because it can be used to integrate multiple types of signals and prevent false starts or spurious induction of gene expression. Normally, this is depicted in a diagram by this half circle. How can you create an AND gate? Well, a classic way of doing this is by using a hybrid promoter. This can be a promoter for a gene which contains DNA sequence elements responding to more than one type of transcription factor or repressor. In this case, we have a promoter which responds both to lactose. Another part of that promoter is responding to the presence of these AHL or quorum sensing molecules. And in a true AND gate, this will only be turned on when both lactose is present, because it will relieve repression from Lac I, and the AHL molecules are present, relieving repression from Lux R. Absence of one of these causes the expression to be limited. So in this case, we have an AND gate. Only when you have those two inputs will you get your output protein. This can also be done using a variety of different sort of combinations of biological activity. Here is one such example, again, of a different combination here, where you'll need two different molecules here, both a tRNA and a transcription factor, to successfully produce this protein. So it is an AND gate. You need both input of A, which here is governed by blue light for this promoter, and input B, which activates this promoter, to create these two molecules as intermediates to activate this final output. Up here, you can actually see the BioBrick accession number if you want to look at this particular construct more closely and get more information on it. Again, this is that same part, just abstracted. Again, writing the relationship between these different parts and genes in this way allows you to build even more complex circuits, since we don't have to worry here about the particular DNA parts. Let's look at some other simple logic gates here. A NAND gate is like an AND gate with an inverter. We'll also look at some OR gates. An OR gate would mean that you'll only get output if you have one or more of those inputs. So a little bit of A, a little bit of B, or a lot of both will allow you to get output. The only time in an OR gate that you don't get output is if you have an absence of both inputs. One easy way to make an OR gate is to simply have the same output under the control of two different promoters. Either promoter gets activated or both of them get activated, you get the output you want. This is, again, another way of depicting an OR gate. A NAND gate will give you output only when you have one or none of the inputs. So this is the opposite of an AND gate. You might already be able to take a guess of how you can take an AND gate and create a NAND gate. You'll simply add an inverter. So we have the same AND gate that we had before. A and B together can add to C, but now C is the intermediate. It's a repressor. It is the part of an inverter, which is going to shut down D. Pretty simple. This is the same approach that can be used to make an XOR gate. So only when you don't have both of or either one of the molecules. This is a little bit more of a complicated one, an XOR gate. This type of a logic gate means that you only get output if you have one but not the other of the input. So no inputs altogether get you no output. Both inputs, like you would see for an AND gate, here give you no output. Only when you have B but not A, or A but not B. These are more complicated, more complex. They could be made through the combination of different parallel circuits. Here's one such example. We can have an AND gate, which is acting as a inverter, while we have other parallel circuits which activate that same final output protein. Again, you could start to see that when you're trying to create more complex logic gates, this can become much more complex. But again, it relies on the same thing of looking at different combinations of promoters and activators and repressors and just stringing them together in the appropriate way. The choices you make here for which activator or repressor, which kind of promoter, will govern the overall dynamic of the system. When you're making more complex circuits, you do have to watch out for crosstalk, for the ability of one part of the circuit to influence another. You want these to be parallel and insulated from each other if possible. Right. In this example, we do have some instances of crosstalk which could muddy the waters a little bit. There are some interesting examples of XOR gates, which I won't go into here. One is from Zurich of 2014, which is worth checking out, a very clever and elegant design. Another one is from the team NTNU Trondheim, which actually uses some anti-mRNAs that can interact with each other and cancel each other out. So again, a pretty clever approach. And finally, there's an example from 2018 that actually uses some enzymatic activities to create an XOR gate based on balancing redox reactions. So, a review of some simple logic gates. An inverter, which is created by adding an intermediate repressor, will flip the behavior of a circuit, whether or not it's just one set of genes or a larger circuit, like taking an AND gate and flipping it to make a NAND gate. An AND gate requires two inputs to give you the output. An OR gate requires any input, and an XOR gate requires only one input. Both inputs would not lead to the output. Now I'd like to introduce some other genetic circuits, and this is where we move from considering the simple logic and the relationship between the different genetic components to now consider how that relationship works when the dynamics of the activation or repression are different. So for the next set of circuits, how fast or slow or strong or weak one of these interactions are will govern the overall behavior. I'd like to start this by showing you a feed-forward loop. In this case, we have a sort of AND gate where you need A and B together to produce C. And A also is producing B. So in this case, input A is activating both target C and the intermediate activator. The difference here is that the speed at which A is activating C is faster than the process by which A goes to B goes to C. That is slower. The special behavior of a feed-forward loop like this means that The only time we'll really get a sustained or strong amount of the output, especially if the end result is this AND gate, is when you have a sustained stimulus. Consider the following. Let's say we had a brief spike in the amount of activator A. It will quickly activate output C, but will dissipate. By the time it's also activated B, the original amount of A is dissipated and it can't combine for the effect at C. So this type of a genetic circuit can be very useful if you find that your behavior is spiking or tripping with noise or low frequency stimulus. If you want to make sure that this is only going to trigger a response when you get a sustained level of whatever stimulating A, you will use a feed-forward loop setup like this. Okay. Here again is another example. If there was a brief pulse of A, the A signal is gone before enough B is produced for A and B to both activate C. So you get no output. Again, by the time B has been produced, we've lost A. Let's say it's been degraded or diffused away. With a longer pulse of A, we've given enough time for B to be produced while A is still present. Now we're going to start to produce that output. So this could be the difference between a brief spike in stimulus versus a more sustained stimulus. This also can be a way of measuring the frequency of a particular stimulus. If we're getting brief pulses at regular intervals, but they are too far apart, if it's too low of a frequency, we won't get production. A higher frequency will allow A and B to be present at the same time, and will eventually build up enough where we will start to produce C. There are some natural examples, which I won't go through here, that resemble a feed-forward loop. A lot of these are tied to cell cycle control. There are other synthetic examples which we can look at here. Again, the basic architecture of this kind of a circuit, you can see that we have input A shown in red. It's activating or leading to production of both the output here and the intermediate activator. Again, a feed-forward loop is where you have A activating B, and activating C, but A and B are both needed to activate C. It's useful for ignoring sporadic or low signals. The final type of circuit I'd like to highlight is known as an incoherent feed-forward loop. This is where input A activates the target C and also activates an intermediate B, which in this case acts as a repressor. Once again, the dynamics between A and B are important for how this works, right? We want to have one branch of this circuit acting faster than the other to get this sort of different dynamic type of response. A neat consequence of this type of circuit is that it will tend to generate a pulse response, or it will detect a relative change in amount or frequency, but not an overall change. So it's a nice way of responding to a stimulus and then equilibrating to those conditions, and then only when something changes actually changing the output. Actually, if you could think about it, there's probably a lot of biological systems and behaviors, maybe even within our own body, that kind of respond this way. You can think about when you encounter a stimulus, but you eventually become desensitized to it. Right? This is the same kind of behavior here created through this incoherent feed-forward loop. Again, this is the kind of behavior we would see if you added a spike, or you actually add, not a spike, but if you add molecule A, even a sustained amount of this molecule will create only a spike in output. Sort of a way of just registering, yes, something has changed. You added A, you're continuing to add it, but after you've done that, the level hasn't changed. The reason why this works is because the addition of A activates C initially, but eventually will repress C through this intermediate repressor. As you can see here, that's what happens at this spike. At the top here, we've created enough B to now repress the signal. Only now when the levels of A change again will we see a change in the output. IFFLs can also act as a bandpass filter. Right? It's kind of interesting where if you have too high of a frequency, it will get that initial spike and then return to a baseline level. If you have too low of a frequency, you'll never be able to really achieve a high level because you'll only your stimulus is too far apart. But on a target frequency, you can actually accumulate here within an incoherent feed-forward loop. So kind of an interesting extra property it has compared to a regular feed-forward. Not only does it discriminate against frequencies that are too low, it also will ignore ones that are too high. There are again some natural examples which I won't go through here. One that might seem familiar to some of you are how neurons actually fire and the way in which they have this sort of negative feedback cycle to make sure they're only sending pulses. Another interesting example of this is actually how plants regulate photosynthesis in response to changes to daylight levels. There is actually an example from iGEM of a team which tried to model an incoherent feed-forward loop, and they did this using protein degradation. This is the William and Mary team from 2017, which I believe actually won a prize for this. And once again, you can see that they were able to achieve this a kind of expected spike and then decline in their output. So these are some exercises that you can consider. How would you take the Lac Operon and change it so it behaves as an incoherent feed-forward loop? I'm going to skip through this here, but just as a way of review, a feed-forward loop requires a sustained level of input required for an output, and an incoherent feed-forward loop generates a pulse in response to fold changes or a certain frequency of stimulus. You can actually combine some of these devices to create much more sophisticated types of outputs, but again, this is a little bit beyond the scope of this video. Thanks for watching.